Hello, welcome. My name is Helena Kshystek, and today I will be talking about my project, which aimed to predict cancer type from microRNA expression data. MicroRNAs are small, processed, non-coding RNAs that are involved in gene regulation, or turning a gene on or off. Changes in microRNA expression has been shown to be associated with cancers. MicroRNAs are being investigated in cancer pathogenesis as biomarkers for cancer and as therapeutic targets. MicroRNAs associated with cancer can be upregulated, which then they are called oncomeres, or downregulated, and then it would be a tumor suppressor mRNA. For my project, I downloaded the raw DEMC2 database, which is a database of differentially expressed mRNAs in different types of cancers. The log FC value is the most attractive for expression for many reasons. For log FC, 0 means no change, a negative value means downregulated, and a positive value means upregulated. 1 unit of the log FCs translates to a twofold change in expression. I also paid attention to the mRNA update ID, which was not unique, if the mRNA was identified in multiple experiments testing different cancers, and also cancer type. Clustering by gene expression is a well-documented approach with a rich literature. According to a paper by D. Hasseler, k-means and hierarchical clustering have the best results. I wanted to apply the approach taken on gene expression and apply it to microRNA expression. I used orange for k-means and hierarchical clustering. Shown is the simple workflow for performing k-means clustering. A fun fact, most algorithms default now to k-means ++. In k-means, an original seed is chosen randomly from within a cluster, and therefore different iterations of the same k-value may produce different results, and neither can be said to be more accurate. However, k-means ++ has an algorithm to optimize the original seed of k-means and therefore is more able to be replicated. Also, in orange, the k-means module will tell you what it actually thinks the optimal k-value is for your data, which is based on the silhouette score. And the silhouette score tells us how well a data point fits into the cluster. So here we have the output of this workflow. You can see you can visualize either using a scatter plot or a box plot. And what I found in clustering was that, especially in k-means, the program was telling me that the optimal amount of clusters was 2, which I really saw only corresponded to upregulated mRNAs and downregulated mRNAs, and this was not very informative to cancer type. I also had difficulty plotting the clusters in a scatter plot when I was only clustering based on one value, which is the log FC value. So here you see clustering for one particular mRNA ID, which was identified in experiments of several different types of clusters, several different types of experiments for different cancers. So here you see not quite what should be the k-means cluster scatter plot. Instead, you have the log FC value here at the bottom and the silhouette score on the y-axis. So saying that cancers, for example, like breast cancer, were more difficult to assign to one cluster or another. Hierarchical clustering worked better for only clustering on the log FC value. In orange, you can adjust the clusters by depth, depth being this range here, and how far down the clusters are actually determined. On the right, I've zoomed in, and you can actually see the cancer listed next to the expression data from which it's derived. You can see that lymphomas and prostate cancers seem to group pretty well together based on this mRNA expression. However, breast cancers, which actually there were very many breast cancer experiments in the database, those are harder to group together, possibly because there are so many values. For future steps, in orange, the k-means module does have a silhouette score, which scores how well the data can be clustered. However, we need a score for how well hierarchical clustering works as compared to k-means. We could use a DBI index to compare the two clustering methods. However, I was not able to find a module in orange for DBI. From my subjective perspective, though, I do think hierarchical clustering was more effective when clustering on only the log FC value. So I took a different approach to analyzing my data after having mixed success with clustering. I thought that if I transposed the table, each mRNA's value could be viewed as a predicting variable to predict the target variable of cancer type. A major problem that I discovered with this approach was that not all microRNAs were identified as differentially expressed in each cancer, and it would be inaccurate to substitute these null values with a 0 value, which would mean that did not change between normal and cancer tissue. Instead, it's an unknown. So this would be, if the data worked, my proposed workflow for predictive modeling using naive Bayes and decision trees classifiers. So you can see I've divided up the workflow into data pre-processing, the naive Bayes workflow, and decision tree. Most of my effort in the predictive modeling went towards data pre-processing. I actually chose KNIME for my predictive modeling because of its pivoting module. After pivoting the data, I was able to group it by experiment and cancer type, which you can see here, and transpose the mRNA values to be columns now instead of rows. This helped. However, there are still many null values where an mRNA was not identified in one experiment but was identified in another. And these null values result in many true negatives when fed through the different classifiers, or in fact the classifiers will just ignore columns with too many missing values. And this leads to very inaccurate classifiers that do not have good predictions. So in summary, how I would improve this project. In terms of clustering, I think a major fault of this project was my choice in data. Instead of going to a database that already identified differentially expressed mRNAs, I should have instead downloaded each experiment and cluster either within an experiment or on different experimental conditions. And from there, I could see if clusters formed. In addition, I would like to find in either KNIME or Orange a DBI index module where I could compare different clustering methods. In terms of predictive modeling, mostly I would focus on, again, the data. A way to improve the data set by decreasing the number of nulls is to impute, or in a way infer, for missing values between experiments of same cancer types, which most easily can be done in R. Thank you for watching this presentation, and here are my references. Thank you.